Very good. So let's walk through how Sanger works. How does Sanger sequencing work? Let's talk about how Sanger sequencing works. Okay, let me zoom out and get this. Okay, let's talk about this. So this here is a deoxynucleotide triphosphate, a DNTP. We discussed this in the first lecture, okay? This is a special, oh, let me spell this correctly. This is a special NTP. It's different because at this position, there is only a hydrogen. So there's no oxygen. So let me ask a question. This one is called a dideoxy. Dideoxy, a DD NTP, dideoxynucleotide triphosphate. So let me ask a question. What will happen if I add a DD NTP to a DNA molecule instead of a DNTP? Yes, very good. Yan Lu, +1 point. It will stop extending. Why? Let's draw it out. So remember how DNA is linked together. Okay. Remember the carbons. This is carbon 3, and this is carbon 5. Okay. DNA is linked at these carbons. Okay. And this oxygen is required to bind the phosphate of the next base. Okay. So if you add a DD NTP to the end of a DNA molecule, it cannot extend. Okay. It cannot extend. Now, this is very, very important for understanding Sanger sequencing. So how does Sanger sequencing work? Okay, so let me explain how Sanger sequencing works. You take a tube, and in the tube, you put DNTPs, a lot of DNTPs, and a few DD NTPs, okay? These go into this tube. So ingredient 1, ingredient 2, 3, you also have a template. So let's add our parasite amplicon. That's about 200 base pairs. Let's also add 1 primer, okay? And they will add a polymerase into this tube for Sanger sequencing. Okay, so here's our template. This is what I call template. This is our 200 base pair amplicon. Okay. Then in Sanger sequencing, they will do a PCR cycle. Okay. So they will denature to anneal. Let's say here's our primer, 1 primer, 3 extend. Okay. Now let's think about the extension when there's a lot of DNTPs and a few DDNTPs. Okay, so the DNTPs will be black and the DDNTPs will be green. Okay, when the polymerase starts writing, most of the bases will be DNTPs. Okay, but at some point, 1 DD NTP will get added. And after that, it cannot extend. Okay. And this is a random probability to add a DD NTP. Okay. This is important. There's a random probability to add a DD NTP. So this PCR reaction will produce, the result of this will be some PCR amplicons that are short, and some will be longer, and some will be longer, and longer, and longer, and most will be middle size, and some will be very, very, very long. And each of these will end with a DD NTP. And these are all different sizes, okay? So this will produce different size amplicons, okay? And in Sanger, oftentimes the DD NTPs will shine colors. They'll have fluorophores. So let's say a DD NTP A will shine yellow, and a DD NTP T will shine orange, and a DD NTP G will shine red, and a DD NTP C will shine blue. Okay, so each of these will shine a different color at the end, depending on the base. Okay. Now, let me ask a question. How can we separate these DNA molecules based on size? How can we separate these DNA molecules based on size? Write it in the chat. Very good. Agarose gel. Okay. So in early Sanger, in classical Sanger, they would take these and they would run a very, very, very long gel. And you would get bands of different sizes that would shine different colors. Okay. And from this, we could know blue was C, 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 C. Yellow was A, 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 A. And red was G, 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 G. And do you see now this? This is our sequence of DNA. So when you do Sanger, here's a question. When you do Sanger sequencing, what do you get back? What does the data look like? What does the data look like? Okay, you get two different types of things. One, you get a .txt file, which is the A, T, G, C, etc. Okay, to N. And this text file comes directly from this process. Okay, so you get a text file with the sequence. Second thing you get, what's called a chromatogram. Okay, a chromatogram looks like this. Chromatogram will look like this, something like this. And you can learn a few things from the chromatogram. The y-axis is the fluorescent intensity, the peak intensity. Okay, so a very, very, very high peak is good data. As you sequence longer and longer and longer and longer reads, the peak intensity will degrade. Okay, so long, we call this long reads. This is like greater than like 500 base pairs. Sequence will degrade in quality. Okay, this is bad data. This is good data. And this will work about 500 base pairs. Okay. So let me ask a question. What if we saw this? What if we saw blue equals G, A, C, and let's do yellow. And this would be T. So my question is, what is the sequence from this chromatogram? Okay, spend 2 minutes, figure out the sequence from this chromatogram. What is the sequence? Take 2 minutes, figure out what is the sequence. Okay, this is kind of a trick. This is kind of a trick. In English, we call this a trick question. Trick question. It means that in English, that means the answer is complicated. Okay, so we would think that, let's see. So here there's a T, here there's a G and a G. Here there is a and an A. But what about this? What about this spot? There's actually both. At that spot, there is both C and there is also T in the sample. What does this mean? Okay, what does this mean biologically? What could this mean? What do you think? You're thinking correctly. You're on the right track, but the answer is not a mismatch. Mutation is closer. Mutation is close. Yeah. So yes, very good. Okay, Piao. Very good. Heterozygote. Very good. Good. Okay. The answer is it's a polymorphism. We call this in English a polymorphism. Or you might have two quote unquote alleles. Okay. So imagine there's a gene X that codes for blue eyes. And imagine if you have a mutation, you can get brown eyes. And because humans are diploid, we have two copies, okay? If you did a PCR and a Sanger sequencing reaction studying this gene, and the human had both copies, let's say got this one from mom and this one from dad, okay, you would see something like this. You would see a polymorphism. It means it's a heterozygote. A heterozygote. Have a good day.
I shall meet death head on. Decade, you've got to get out of here. Beware foul demons and beasts. There are many foes here. The time has come to cleanse my horde. This monastery reeks with evil and corruption. Oh, I hate staining my hands with the blood of foul sorcerers. Eternal suffering would be too great for you, Diablo. Let the gate be open to die. I shall meet death head on. I shall meet death head on. Decade, you've got to get out of here. Beware foul demons and beasts. There are many foes here. The prime evils are no more.